Hello friends, welcome. We're talking about restriction enzyme. Restriction enzyme treat for molecular biology. First of all, what is restriction enzyme? It is a simply type of enzyme and restriction enzyme, those enzyme act as endonuclease. The term of endonuclease. So here, what is the endonuclease? See, DNA technology totally depend on ability to cut DNA molecule at the specific site with the restriction endonuclease. See here I talking you. It's simply way to explain you what is meant by endonuclease. This is a circular DNA. It is also called as linear DNA. This DNA, this is a restriction enzyme. Restriction enzyme, it is a cut DNA molecule. Here, this dot, this is a restriction site. Which one site here? Restriction site. It is a nuclease enzyme. See, restriction site, DNA cut molecule on the specific site. Who's cutting here? DNA molecule or DNA sequence? Restriction enzyme. Restriction enzyme with the restriction enzyme using the cut DNA molecule at the specific site, and it gives DNA fragments. Here, DNA fragments, and this is the definition of endonuclease. DNA technology totally depend on ability. Endonuclease or restriction enzyme have a ability to cut DNA molecule at the specific site with the restriction endonuclease. See, the types of restriction endonuclease, these are three types of endonucleases. Type first, type second, and type third. See, first, see what is the endonuclease and what is kind of endonuclease. Restriction enzyme. Here, restriction enzyme is most important type of endonuclease that are present inside our body, and also we use this term manipulate molecular biology studies, because it is using the breaking of DNA. But restriction applied, restriction are cleavage enzyme. See, one by one types of endonuclease. Type first one, type first. But first of all, I'll tell you enzymes. First enzyme, see, I write here enzymes. I write here enzymes. The first, hemophilus influenza. Hemophilus influenza, it is a enzyme or it is a gene producing organism. This is a producing organism, and this enzyme, it is using a short form HIN. Second one, Escherichia coli. This is also producing enzyme, and it is also called as E. coli. It is a shortly using this word E. coli. or eco R1. These are genes. Or enzymes. See, first type. One more genes here. Eco R1, HIND3. It is also called as HIND3. SAL1, col E1. See, one by one types. First one, endonuclease type first. Complex, it is complex nucleus. Here presence of genes eco R1, this is the first point. Second point here, ATP and Mg++ are required. ATP means adenine triphosphate. It is require energy. ATP means is require ATP. Third one, modification. There are three types of endonuclease type. First, there are three types. In the presence of three types, modification, restriction, and third one, specificity. Fourth one, here 15 base pair length. How many? 15 base pair length. Fifth one, methylase. This is also enzyme. This one is enzyme using here methylase. Methylase is a cleavage site. And here 1000 base pair. Last one point here, nucleotide sequence here, presence of thymine, cytosine and adenine. This is a nucleotide sequence base pair. Second type second. See, it is a first one. It is simple chain enzyme. It is a simple enzyme. It is simple enzyme. Second one, it is specific nucleotide. I told you about nucleotide sequence. Nucleotide sequence means adenine, this is A, guanine, adenine, G, thymine for T, cytosine for C, and last one is uracil for U. Uracil is used for only RNA, and it is replace of thymine, and this one is using for DNA. Let's clear? Okay. See, specific nucleotide sequence hexagonal, butanol, tetra. These are nucleotide sequence. Third, Mg++ as a cofactor DNA sequence left to right. Here DNA sequence left to